Hi, I'm Richard Scheines from Carnegie Mellon University.
And I'm Greg Cooper from the University of Pittsburgh. And today we're going to be speaking with you about causal discovery from big biomedical data.
In today's talk, we'll talk about the purpose of causal discovery algorithms. We'll introduce you to the methods for discovering causal networks, and we'll talk about the available resources for discovering networks. So science has many reasons to look at causation. We want to understand the mechanisms of things. We want to predict the results of interventions. We want to control events if we possibly can. Some examples are discovering the clinical effects of a new drug, determining the genes and cell signaling pathways that cause breast cancer, and uncovering the mechanisms of a recently mutated virus. So why have the methods for graphical causal discovery been advancing so rapidly? Well, there's two really big features. One is there's been amazing algorithmic advances over the last several years. And two is there's way more availability of big biomedical data than there used to be. In fact, over the last 25 years, there have been substantial advances in this field. We have now figured out how to mathematically represent causal networks. We've specified lots of different kinds of discovery algorithms for finding networks, and we've now been able to apply these methods to biomedical data. The variety, richness, and quantity of biomedical data have been increasing very rapidly. We have high-throughput molecular data. We have clinical EMR data. And we have population health data from social media and mobile sensors. And the appropriate analysis of these data has great potential to advance biomedical science. So what's the basic discovery workflow when you're trying to get causation out of data? Well, we have prior knowledge, we have data, and we use those in causal analysis to produce causal networks, which we'll explain later. The data can be both observational and experimental. And the causal networks generate for us hypotheses that we can then test with future follow-up experiments or other kinds of work. And the main goal of the search methods is to really suggest causal hypotheses, not to exactly figure out what particular hypothesis is the correct or true one. So the causal hypotheses we generate lead to experiments we can do, which produce more data and more prior knowledge. And so it's a circle we continue to iterate on forever. So let's give an example of this technique and so you can get a sense of what we're talking about. For flowers, it's important for people who produce them to figure out how to get them to flower at a particular time. And so there was this question that came up with this particular flower: which are the genes that regulate flowering time? And so the researchers who actually were working in Europe took 47 plants and they looked at all the gene expression profiles of 4-day-old seedlings and then try to figure out which of those genes had an effect on flowering time. They measured 21,000 genes, and then they took the observational data they got from a microarray, and they did a causal analysis, figured out what networks would actually fit the data, and then selected the genes that reliably caused the most substantial changes in flowering time according to those networks, got candidate gene regulators, and then actually put experiments together in which we tested whether or not those regulators as candidates really did influence flowering time. So what happened? Well, we got 25 genes that we took as candidates, and we thought which of these would reliably cause substantial differences in flowering time. Well, luckily 5 of those 25 were already known as regulators of flowering. So far, so good. And then 13 of the 25 that were not known regulators, we had mutant seeds, and we tried to test which of those would actually delay flowering or increase flowering. So we basically had a bunch of data we took from microarray measurements. We did an analysis with the algorithms we'll explain later. We got a candidate gene regulator set. We did experiments to follow up. And the experiments were basically seeds, right, with Columbia Arabidopsis thaliana wild type and homozygous insertion mutants, which we obtained from the Nottingham Arabidopsis Stock Center. Those seeds were plated, put on a medium, stratified for 2 days, and then grown on plates for 10 days. And then they were transferred into soil in confined growth chambers. We measured the flowering time, and the seed types yielding 4 more plants were then considered for analysis. So there were 9 seed types, each with a single gene insertion that yielded 4 more plants. And of those 9, there were 4 that had a statistically significant shorter mean flowering time than the control, wild-type plants. So overall, 4 out of 13, which is about 31%, gene-altered seeds produced significantly shorter flowering time than did wild-type plants. And that really is, for us, experimental efficiency of very, very high sort. Okay, so that's an example using flowering as the target, and you can see how it works from the high level. And now let's drill down a little bit into what were the components that we needed to actually figure out in order to get that to happen. So there are really three different things we have to be able to do in order to actually perform this kind of work. First, we have to figure out a way to represent causal models. And second, we have to evaluate these models with data, so we see which models fit the data or explain the data better than which others. And we also have to be able to search among a very, very large space of models and determine which of those models in that huge space are the ones that actually fit the data well enough to consider as candidate causal hypotheses. So representation starts out with a directed causal graph or a causal network, where we have nodes that represent the variables. So in this example here, we have a node for television watching, which we say could either be low or by high. We have a node for diet, which is caloric intake, and we have a node for body mass index. The edges, or the arcs, in these models represent direct causal influences relative to the set of variables we have in the model. So in this case, TV has a direct influence on body mass index relative to diet, and it has a direct influence on diet relative to BMI. Diet has a direct influence on BMI relative to TV, and that's the only set of variables we're actually considering, so this specifies our best guess about what the causal relationships are. Now, if we change the set of causal variables, so if we add another variable, exercise, for example, then the relationship between TV and BMI, which was direct in the previous model, is no longer direct. We think that pathway goes through exercise, and so we take away the edge from TV to BMI. So there's no sense in which an edge is an absolute claim. It's relative to the set of variables one considers. The probabilistic relationships really record for each variable its probability distribution conditional on every possible value of the parents or the direct causes of that variable. So for example, for exercise, we need to say what the probability that exercise is low, given that its parent, direct cause, TV, is equal to high, and we also need to say what the probability of exercise equal low is for its parent, TV, equal low. Another way to parameterize probabilistic relationships in these variables is to express each variable as a function of its direct causes plus some stochastic noise. So for this example, we might say diet, which is caloric intake per day, is 1,800 calories per day plus 1.7 times the number of minutes of TV watched plus an epsilon sub-diet term, which itself is normally distributed with a mean of 0 and a variance of 200. The final piece of the representational puzzle is intervention, which is crucial for anything that talks about causation. So take this example again and imagine an experiment in which we intervened on people's diets. We might get participants to volunteer, and those participants might agree to be randomized to one of two conditions. In one condition, they would be given a diet that was 1,500 calories a day, for example, and in a different condition, they might be given a diet that had 2,500 calories a day. Now, because they've agreed to participate in this experiment and let us control their diet, whatever TV they were watching, and whatever influence that was having on their diet previous to the experiment, is surgically removed, and now the randomizer is the only thing controlling diet. So finally, for us to actually evaluate models, we have to have some way to connect the structure of the model to testable constraints we could look at in data. And so the causal networks, which are entirely qualitative representations of causal structures, have been connected to an entirely qualitative testable constraint in data that is conditional independence relations. So the notation we use for conditional independence, as we show here, is that X is conditionally independent of Z on Y, or X is independent of Z conditional on Y. And if we're going to go the other way, if we're going to search for causal models from data, then we have to figure out some way to start with predictions that we could test and then see which models might actually generate those kind of predictions. And so if we start with conditional independence, something we can test from the data and make statistical decisions about, and we have a discovery algorithm, that algorithm could generate information about the causal structures that might have generated those testable constraints. And what we get when we do that kind of a thing for conditional independence relations is not a single causal model, but we get an equivalence class of causal networks, all of which entail the exact same set of constraints, namely the ones we actually observed in the data. So the types of data we look at include experimental data sets, where we control or manipulate some of the variables and observe others, observational data sets, where we don't manipulate any variable, we simply observe, and then data sets that are a combination, where we have experimental and observational data. We're going to focus on observational data for the moment, and we're going to go through in a little more detail the algorithms and how they actually work on a very simple toy example. And for this part of the presentation, let me turn it over to my colleague Greg Cooper from the University of Pittsburgh.
So let's suppose that we're interested in evaluating whether or not Y causes Z. The traditional method for doing this would involve identifying all confounders of Y and Z, and here those are just being represented by a single variable C, but in general C would contain a set of variables that include all the ways that we can think of that Y and Z might be caused by some common variable. We want to measure these so that we can control for them and by conditioning on them, and then the residual statistical dependency between Y and Z, once we control for C's influence on them, would be the causal effect of Y on Z. A concern with this approach, however, is what if we haven't measured all of the confounders of Y and Z? That is, there's some hidden confounders, and here those are being represented by the variable H. In general, there could be many such hidden confounders. So I'm going to talk about some methods that address this problem and more general types of problems when there can be hidden confounders that we would like to be aware of and have the methodology control for. There's a constraint-based method that's been explored extensively. There's also Bayesian methods for learning graphical causal models from data in light of potential hidden confounding. And there's some other methods. But I'm going to focus on the constraint-based methods because it provides the clearest illustration in a short amount of time about what is going on in terms of causal discovery with these types of methods. The constraint-based method can be summarized in two steps. The first one is to determine the constraints that are holding among the nodes that are being measured. And we're going to focus on independence conditions, which may be obtained by doing, for example, a chi-square test to find that two variables are independent given conditioning on another set of variables. The second step is to use those patterns of constraints to narrow the causal possibilities that are consistent with them. So let's look at a hypothetical example of the constraint-based method. Suppose we have three binary variables, x, y, and z, and the following is known. X occurs before y, and x occurs before z. So, as a particular example, X might be gene sequence information, for example, gene mutation data for some particular gene X. Y could be gene expression data for some particular gene Y, which could be different than X, of course. And then Z could be the presence or absence of a disease, so a binary variable. The question then is, again, does Y cause Z? Does gene expression cause a disease for some particular gene Y? So let's consider a hypothetical example of the constraint-based method. Suppose that statistical testing on a large data set yields the following four constraints that are found in the data. Variable X and Y are dependent, variable Y and Z are dependent, variable X and Z are dependent, and we find that X and Z are independent given Y. So let's consider the consistency of these constraints with respect to a number of causal networks that the algorithm is going to search over. And I'm going to give you a little bit of time to consider these as we go through them. So the first model, let's suppose that we consider in our search is that X causes Y, and there's no causal relationship between X and Y and Z. Do you think these are consistent with the four constraints shown above? Well, we have that X causes Y, so we'd expect the dependency there, and we find it in the data. We also find in our dependencies above that Y and Z are dependent. However, this causal model really doesn't support that, and I think your intuition would be supportive of that there's really no way for them to be dependent given this causal structure. So that one is not consistent, and therefore we would conclude that this is not a model that evaluates as consistent with the four constraints. What about this possibility? Well, similarly, we have some of the dependencies that we find in the data, Y and Z, but we don't have dependencies between X and Y and X and Z that this causal structure supports. However, we find those dependencies in the data above. So this one we wouldn't consider as consistent with the constraints. What about this one where X causes Z and Z causes Y in the causal structure? Well, we have the pairwise dependencies that we found in the data above. What about the independence of X and Z given Y? Well, here we find that X is causing Z and Z is causing Y. If we conditioned on Y, we wouldn't expect that to lead to an independence between X and Z. We would still expect that X could have a causal influence on Z. So that independence found in the data does not hold, and we would not consider this one consistent with the data. What about this one where X causes Y and Y causes Z? Well, here we again have the pairwise dependencies, X and Y, Y and Z, and then we'd expect X and Z to be dependent through transitivity through Y. What about the independence of X and Z given Y? Well, yes, since X only causes Z through Y, if we conditioned on Y, we would expect in the data for X and Z to be independent. So we give that one a green check. What about this possibility? Well, we don't have time to go through this in detail today, but it turns out that this is also consistent with the four constraints above. And another causal structure that's consistent is shown here. So it turns out that there are actually 90 additional causal networks on these three variables that we could consider, but because of time, we're not. It turns out that none of the other 90 actually satisfy the four constraints shown above. So the only three that do satisfy the four constraints are the three here on the right that are shown. And notice what's consistent among these three. They have Y causing Z, and there being no hidden confounder of Y and Z. So that means that we can estimate the causal effect of Y causing Z from the observational data. This slide just summarizes the points that I just made. So in summary, for the constraint-based causal discovery method, what it does is it reduces a large number of causal network possibilities to just those causal networks that are consistent with the constraints found in the data. And it looks for the causal relationships that are similar or invariant across those networks. In the example that we saw, it was that Y causes Z, and they are not confounded. They have no hidden confounding. This method, although we've just considered it for three variables, generalizes to thousands of variables, for example, gene variables, and thousands of samples. Let me now consider a real example of using this constraint-based method that's taken from the paper shown here, titled Epigenome-Wide Association Data Implicate DNA Methylation as an Intermediary of Genetic Risk in Rheumatoid Arthritis. The goal of this paper was to determine whether specific sites of DNA methylation causally influence acquiring rheumatoid arthritis in humans. And they did use the three-variable approach in much of their analysis. In particular, they used observational data of 354 positive rheumatoid arthritis cases and 337 controls from patients in Sweden. X represented SNP measurements from the Illumina human hap chip. Y represented methylation positions measured with the Illumina human methylation 450 array. And the Z variable represented rheumatoid arthritis status, either present or absent. If we consider this analysis problem in terms of our general causal network workflow, we see that the X variables can represent many different SNP array measurements. The Y variables represent many different methylation array measurements, and Z always represents rheumatoid arthritis status. These then become input to our causal analysis, and for each type of X, each type of Y, and for Z, we do the kinds of causal network analysis that we saw earlier in the hypothetical example, where we find the networks among X, Y, and Z that are consistent with the constraints found in the data for that X, Y, and Z. We then select those X, Y, Z triplets that have constraints supporting that Y is an unconfounded cause of Z. So then those become candidate methylations that causally influence rheumatoid arthritis. In particular, those Ys in those triplets become those candidate methylations. So let's look at the candidate differential methylation positions that causally influence rheumatoid arthritis that were found in the results. There were 535 SNP DMP pairs in the MHC region that satisfied the 4 needed constraints on X, Y, and Z. These pairs consisted of 264 unique SNPs and 9 unique DMPs, methylation positions. These 9 differential methylation positions are then candidates as causes for rheumatoid arthritis. So they weren't able to perform experiments, at least as reported in this paper. However, they did do a validation experiment to look at a different set of patients to see if there was consistency in the results they obtained with that additional set. So, in particular, they evaluated a separate set of patients to assess whether the methylation differences also occurred in them in these 9 differential methylation positions. They used 12 rheumatoid arthritis cases and 12 controls. They then extracted from these monocytes from the whole blood using flow cytometry, and they measured the DNA methylation levels using the Illumina 450 methylation array. And they compared the methylation levels for the 9 DMPs in the cases versus these matched controls. The results that they found are as follows. All 9 of the DMPs that were found by the causal analysis using a method that's very similar to what we illustrated in the hypothetical example showed methylation changes in the same direction as the original analysis. So there was this type of validation. 3 of the 9 were statistically significant at a p-value less than 0.05. 1 had a p-value of 0.06, and 1 had a p-value of 0.11. And recall that this is a small sample size in this validation set, only 24 total cases. So the conclusions then are, although the validation experiment had a small number of samples, overall the results are consistent with the original study. 30% of the DMPs in the validation experiment had statistically significant changes that were in the same direction as the original study. To read more details about what we've been talking about so far in terms of causal representation, causal model evaluation, causal model search, we recommend this particular paper, which can be obtained at the location indicated at the bottom line here. It talks about more details, an additional level of detail about the computational causal discovery methods, in particular for systems biology, and gives a few more examples of applying those methods to real data. In the remainder of this presentation, I'd like to give you a brief overview of the Center for Causal Discovery. The main goal of the center is to help biomedical scientists discover valid, novel, and significant causal relationships in big biomedical data that will lead to new insights in health and disease. The center is an NIH Big Data to Knowledge Center of Excellence. It's a collaboration of more than 50 investigators at the University of Pittsburgh, Carnegie Mellon University, the Pittsburgh Supercomputing Center, and Yale University. We're developing and deploying free open source software for causal analysis of data, like the analyses that we've talked about so far. There's several different forms of the tools. One is the Tetrad desktop application that makes it easy to analyze data with a graphical user interface. There's also a web-based application that allows you to use a browser to connect to the Pittsburgh Supercomputer Center to analyze big biomedical data. We, in addition, have Java, Python, and R versions of key algorithms that allow you to then use those algorithms within your own code workflow. Also, there's training in causal modeling and discovery. In particular, tutorial videos and training modules are available on the website shown above. And the center is investigating 3 driving biomedical projects. The reasons for these projects are to evaluate the usefulness of the causal discovery algorithms on these 3 problems, and also to drive further development of the algorithms. Here's the 3 projects with a bit more detail. One involves discovering cell signaling networks in cancer. Another is discovering the mechanisms of disease onset and progression in chronic obstructive pulmonary disease and in idiopathic pulmonary fibrosis. And a third project is involved in discovering the functional or causal connectivity of regions of the human brain from fMRI data. In summary, discovering causal relationships is at the core of biomedical and clinical science. Graphical methods exist for representing and searching for causal relationships from data, including purely observational data, as we've seen in some examples. And the Center for Causal Discovery is making tools available for discovering such causal relationships in the form of causal networks from large biomedical data sets. We'd like to acknowledge the National Institutes of Health for their support of the Center for Causal Discovery. Thank you. And if you'd like additional information, again, please see our website shown here.